Welcome to the second half of the Chapter 21 Part B, where we're going to focus on cellular immune responses. So the responses that are kind of regulated and guided by the T lymphocytes, the T cells, which is what you guys see here. These are some T cells attacking a cancerous cell. So T cells really are about fighting against antigens that are inside other cells, and so they require the antigens to be presented, and we're going to talk about that in just a second. There are some T cells, we call them cytotoxic T cells, that will directly kill other cells, and this is really why it's a cellular immune response, because the cells themselves are destroying the infected cells. There are other T cells, helper T cells, regulatory T cells, that release chemicals and play roles in activating and regulating other parts of the immune response. And we'll look at those kinds of different roles of the different types and populations of T cells. So we do find that we have two populations of T cells. Whether they're a CD4 cell or a CD8 cell depends on which surface receptor that they display and whether that surface receptor responds to class 2 or class 1 major histocompatibility complex protein. So the CD4 cells are the ones that become helper and regulatory T cells. They have receptors for the class 2 MHC proteins, and the helper T cells will go and activate other B cells, other T cells. They'll go activate innate macrophages, and so they'll direct kind of both the adaptive immune response and, when they activate macrophages, the innate immune response. The regulatory T cells are about moderation, so making sure that the immune response is kind of the appropriate strength and not occurring inappropriately, helps prevent autoimmune diseases, things like that. These CD4 cells can also become memory T cells as well. So just like with our B lymphocytes, we have T lymphocytes that will remain as memory cells that will induce a stronger, faster secondary immune response upon second exposure to a specific antigen. The CD8 T cells are the ones that become the cytotoxic T cells, the ones that actually go and kill other cells. And they have receptors for the class 1 major histocompatibility protein, and they will go and attack these other cells that have these antigens that the body recognizes as foreign. The CD8 cells can also become memory T cells. So again, this idea of memory is a very, very powerful advantage to our adaptive immune response. So if we just kind of look at kind of how this kind of occurs, here you can see here's our immature lymphocyte in the red bone marrow. It then seeds to the thymus, where it will then display the receptors either for a class 2 MHC protein if it's a CD4 cell, or a class 1 MHC protein receptor if it's a CD8 cell. If we look at the CD4 cell first, you can see it gets activated when it sees an antigen. It has to be presented by one of these antigen presenting cells, and then once activated, it will go through clonal selection and make lots of copies of itself and proliferate, and then differentiate into either a helper T cell or a regulatory T cell. If we look at the path of the CD8 cells, they respond to antigens that are displayed on class 1 MHC proteins, and then they will differentiate and go through clonal selection and make lots of copies and proliferate and differentiate and become the cytotoxic T cells. And both the CD8 cells and the CD4 cells can become memory cells that are already primed for these specific antigens and ready to kick in that much stronger, faster secondary immune response. So you'll see that these major histocompatibility proteins are really, really crucial in activating cellular immunity and activating these T lymphocytes, because the T cells will only respond to antigens or the antigen fragments, the antigenic determinants that are displayed on the surfaces of other cells. So unlike antibodies that are made by B cells that can respond to, say, perhaps a part of a pathogen that's kind of just floating in the fluid in the plasma, T cells can't do that. They can only respond to antigens that are displayed through these MHC proteins on the surface of other cells. So not only is that how we activate the naive T cells and turn them into immunocompetent primed T cells, it's also incredibly vital for the T cells themselves, once they're in their effector stage, to actually do their job. So we do have two types of MHC proteins. So the class 1 MHC proteins, every single cell in your body will display except the red blood cells, and they will activate the CD8 T cells, as we saw in that previous figure. And then the class 2 MHC proteins are displayed only by the antigen presenting cells, the dendritic cells, B cells, and macrophages, and they will activate CD4 T cells. And so that's what we're kind of looking at here. So let's look at class 1 first. All nucleated cells will display a class 1 MHC protein. When they display this class 1 MHC protein with an antigen bound into it, then they can activate these naive CD8 T cells and cytotoxic effector T cells. If the cell that has this class 1 MHC protein is one of those antigen presenting cells, it is basically telling the immune system that, hey, I'm a self cell, I'm an antigen presenting cell that belongs to this body, but I have something foreign and this is what it looks like. Please go and kill and find other cells that have this particular antigen, because this antigen is bad. If the cell is a cell of the body that's not one of the antigen presenting cells, if it's an infected cell that is displaying this foreign antigen, the MHC protein will tell the immune system that the cell itself belongs to the body, but that it has been infected, or it's become cancerous, or there's something wrong with it. And it's basically saying, hey, I'm an infected cell, I should be killed and destroyed. And then the cytotoxic T cells will do so. For the class 2 MHC proteins, these are only on these antigen presenting cells. So the dendritic cells, the macrophages, and B cells also display these class 2 MHC proteins. They go and activate the CD4 T cells that then differentiate into helper T cells. And what they're kind of telling the immune system when they have an antigen and are displaying an antigen, the MHC protein says that this antigen presenting cell belongs to the body, but it's captured something foreign, and this is what this foreign antigen looks like. Let's band together and go fight it, because that's what our immune system is. They're like the warriors of our body. So that's kind of really how this whole idea of antigen presenting works. In order to get the T cells activated, not only does the antigen have to bind, but we also have to have this co-stimulation. So the T cell itself has to recognize the antigen that's being displayed on one of those MHC complexes, but there are like these little co-stimulatory proteins on the antigen presenting cell that also have to recognize the T cell, and so it's kind of this kind of double bind, co-stimulation. And if that doesn't occur, say the T cell recognizes the antigen MHC complex, but the co-stimulatory protein on the antigen-presenting cell doesn't recognize the T cell, then perhaps we might get an inactivation or an activation of some T cells that are really not appropriate and could lead to autoimmune diseases. So both of these things have to happen, both the antigen presentation and this kind of double recognition in order for clonal selection and proliferation of our T cells to occur. So that's kind of what that looks like. So here's a bacterial antigen. It's been engulfed by a dendritic cell, and that dendritic cell is displaying a class 2 MHC protein. The dendritic cell will kind of process the bacterial antigen and then display that antigen on the MHC2 protein. And here you can see here's this little co-stimulatory molecule. And so it's this idea of double recognition. Not only does the T cell have to recognize the antigen on the APC, this co-stimulatory molecule on the APC has to recognize the naive T cell. And if only one of these happens, if this here recognizes this here, but this here doesn't recognize this here, then the T cell will not be activated. So we must have that co-stimulation. Once we have that co-stimulation, then we can get the formation of all of these clones that are ready to go and do their job and fight against this particular antigen. Since this particular image is looking at CD4 cells, some of them are going to become helper cells, and some of them are going to become memory cells. The same process would happen with the CD8 cells, except this would be maybe, say, a liver cell displaying a class 1 MHC protein, or a dendritic cell displaying a class 1 MHC protein, either or. Takes about a week, really, maybe a little less than, for T cells to kind of kick in and do their job. But as the kind of antigen is destroyed and as the body kind of cleans itself up, the effector activity, the amount of T cells that are doing their job, tends to also wane as it is needed less, right? If you're we start off with 50,000 cells that are infected, and a week later, if 25,000 cells of those are infected because we've already gotten rid of half of them, then we don't need as many T cells around. Anywhere between 7 days and a month, the T cells themselves will go through apoptosis, except the memory ones, because they'll remain. But the reason we don't want the kind of activated T cells, especially those cytotoxic T cells, around, is that they're cytotoxic. They're toxic to cells, and they could potentially damage the body. And since many of these helper T cells in particular also work with the innate immune system and enhance inflammatory responses, then you get this kind of prolonged inflammatory response, which is also not something that we want to see. So the T cells, the effector T cells, will go through cell suicide after they're kind of no longer needed. The exception to that is those memory cells that will remain and circulate through the lymphoid organs and the plasma, ready to kick in with those secondary responses. So cytokines, they seem really complicated, and your book talks a lot about them and gives you lots of different examples, and it's crazy complicated sounding, but all you have to remember is all cytokines are chemical messengers. They're just little tiny chemicals that are secreted by different cells of the immune system that help B cells and T cells develop properly, that help them differentiate into effectors properly, and generally just kind of regulate responses in the immune system. Cytokines are how the immune system talks to other parts of the immune system. A really good example here is interleukin-1. That's one type of cytokine. It gets released by macrophages that will then go and bind onto T cells that will then release interleukin-2 and also tell the T cells to and other body cells to go and make more receptors for interleukin-2, because interleukin-2 is a growth factor that is used by T cells to grow and develop and differentiate. So another example is gamma interferon. It is released by T cells, helper T cells, and binds onto macrophages and makes them more able and better able to phagocytize pathogens. So that's all that cytokines are, just little chemical messengers, and how parts of the immune system talk to other cells of the immune system. So let's look at the three populations of effector cells. The first of which we're going to look at are the helper cells. These are really incredibly vital to the functioning of your immune system, because they play roles in both humoral and cellular immunity. So they can activate T cells and B cells. They can kind of turn on the proliferation of both T cells and B cells, and these helper T cells secrete a whole host of cytokines that recruit other immune cells like macrophages, like dendritic cells, into the immune response to kind of amp up the immune response. And so if you damage your thymus such that you're not developing T cells properly, not only would you lose your cellular division of adaptive immunity, you'd lose your humoral division too. Even though you'd be developing B cells in the bone marrow, they would not be able to activate properly without these helper T cells. Let's take a look at that. So here, let's look at humoral immunity. Here's our little helper T cell, and here is a B cell that has an antigen that it's processed, that it's engulfed, and some of the cytokines, the chemical messengers created and secreted by this helper T cell, help the B cell activate into a plasma cell that could then eventually secrete antibodies that could fight against this particular antigen. And so if you do not have helper T cells, you're really going to lose both your humoral and cellular immunity. Here is a helper T cell that's helping to activate a CD8 T cell to become a T cell. It really is very vital that we get these helper T cells developing in the thymus. The cytotoxic T cells, that's what you see here, these are the ones that actually go and kill things. These are the warriors, really, the knights, if you would. They are found in the blood, in the lymph, in the lymphoid organs, in all sorts of different body cells, and they basically roam through the body, knights on a quest, looking for cells that are infected. It could be infected with a virus, it could be infected with a parasite or bacteria, it might be a cancer cell, like you see here, that's just going through some exponential growth, or it could be something foreign, like if you get an improper blood transfusion or if you get a transplant. That organ would have MHC proteins that the body would not recognize as self, and so these T cells could potentially attack them. And they induce apoptosis in these infected cells in one of two ways. The first mechanism is just by release of two chemicals called porphyrins and granzymes, which I've got a figure to show you in just a second, so you can see how they induce apoptosis in the infected cells. And then cytotoxic T cells also will bind specific receptors on the target cell, in the membrane of the target cell, and induce apoptosis, cell lysis. So let's look at this porphyrins and granzymes process. So here's your cytotoxic T cell, and it has recognized a target cell. It says, oh, this cell is displaying a foreign antigen, and I should get rid of it. And so it kind of really tightly binds onto that foreign cell once it has, for instance, recognized the foreign antigen on the MHC1 protein. And then the cytotoxic T cell will basically via exocytosis release these porphyrin. Does that kind of sound like perforate? Because that's exactly what they do. The porphyrin molecules basically create a hole. They puncture the cell, the target cell, and then these granzymes are enzymes that will essentially trigger apoptosis and start destroying the cell from the inside. And then once it has induced apoptosis in the target cell, the T cell will detach and go and look for another infected cell to kill. The last of the populations of T cells are the regulatory T cells, and they are particularly important in preventing autoimmune diseases. So if your regulatory T cells don't develop properly, then any lymphocytes that are self-reactive, that react to the body's own cells, might not be taken out of commission like they are supposed to. Regulatory T cells can directly contact with self-immune cells, or they might secrete some inhibitory cytokines, inhibitory chemical messengers to these self-reactive lymphocytes, and we want that to occur. We do not want our body's own immune cells attacking parts of the body's cells, other parts of the body. Rheumatoid arthritis is an autoimmune disease where there are immune cells that attack the joints. Multiple sclerosis is an autoimmune disease where the body's immune cells attack the myelin that we find around our neurons. And so anytime the body's own immune cells are attacking the body's own cells, that's an autoimmune disease, an autoimmune reaction. And that's really the job of the regulatory T cells to prevent. And so if someone has an autoimmune disease like MS or RA, then it's a problem with their regulatory T cells. This is just a nice kind of overall figure of kind of the two branches of the adaptive immunity, as well as a little bit here in terms of the innate immunity. So if we look at innate immunity, we've got the surface barriers like your skin, and if they're breached, then we have things like complement, like you see here, or phagocytosis, or fever, or inflammation. And then we really have these adaptive defenses that are more specific, but take a little bit of time to turn on, whereas these are less specific, more general, but are kind of there a little bit more immediately. If we look at the humoral immunity here, we get the antigen that gets activated by the B cells. B cells activate and become plasma cells that will secrete antibodies, which will inhibit this antigen. Some of them will become memory cells for a secondary response. And then if we look at the cellular immunity, here we have an antigen-presenting cell that's been infected, so it will display that antigen to both the CD8 T cells and the CD4 T cells. The CD8 T cells will become memory cells and cytotoxic cells, which go back and kind of kill cells displaying the antigen. They also activate CD4 cells, which become memory cells and helper cells, and regulatory T cells, which can go and activate natural killer cells, macrophages, and also go and co-stimulate and co-activate the B cells so that they're doing their job properly. And so again, here's this helper T cell that's both part of humoral and cellular immunity. The helper T cells in particular and the antibodies that can turn on complement kind of overlap with the innate defenses. So it's kind of a nice little summary figure. There's also a nice little table here from your book that lists the different kind of cells of the adaptive immune response, the different molecules that are made by or turned on by the adaptive immune response, and also what they do. So also a nice little kind of summary table to look at. And that's the end of chapter 21.